Well, thank you very much for these great presentations, Shabnam and Lynn. I think you've really very comprehensively covered the topic of safety evaluation early and late in clinical trials. And Lynn, thanks for addressing really some of the most important subpopulations, which are obviously addressed in regulations and requirements as well. So I'm going to move on to questions, and there are many of them, excellent questions from the audience. I'm going to start off with Shabnam. Shabnam, there's a question for you. What role do investigators play in ensuring patient safety during clinical trials, and what measures are in place to monitor and report adverse events? So this is about the role of investigators in safety.
Okay, yeah, thanks, Leonard, for that question, and thank you for the audience as well. So what role do clinical investigators play? Yeah, that's a great question. Investigators play a key role, as you heard during the presentation, especially when it comes to reporting SAEs to the sponsor. Keeping in mind the exceptions that we discussed in the talk, investigators are involved in determining if the AE is serious, and you could refer to the list of questions to ask yourself about what constitutes the AE as serious. There were, serious is a regulatory definition, so it's clearly defined there. As investigators, it's you're required to provide a causality assessment for each of the SAEs reported to the sponsor, and that really helps the sponsor because the investigator's view is important when considering, when assessing the safety of the drug and determining whether to report the event eventually to FDA. It's the investigator who monitors the subject's response to the drug and is knowledgeable about the subject's clinical state. They know the medical history and the concomitant medications that the patient was on, and so they may be especially sensitive to distinctions between events that could be related to the drug versus those that might be due to the underlying disease process, for example. Over, Leonard. Thank you.
Thanks very much, Shabnam. I think obviously emphasizing very important points that probably nobody understands the adverse event as well as the investigator saw it. So obviously a very important source for information from us. So Lynn, I have a question for you. Interesting question which has come up here. If a trial protocol excludes pregnant women and a pregnancy occurs during a trial, what are the regulations or best practices for follow-up on the baby, including the length of follow-up? So what happens if somebody gets pregnant during a trial? Any thoughts there?
Thank you, Leonard, for that question, and thanks for the question being asked. So even when it's appropriate to exclude pregnant individuals from clinical trials, and I hope I've convinced you that we should move away from the automatic exclusion of pregnant individuals from clinical trials. But even when they are appropriately excluded, pregnancies happen. Not every contraceptive measure is 100% effective. And so I think the question is a very good one. Part of the importance is not to automatically remove the patient from the clinical trial, stop all follow-up, and lose that patient from potential important follow-up measures. So one of the things I think I mentioned earlier is the first thing that should happen is that the patient, if it's a blinded controlled trial, you have to break the blind on that patient because you need to know if that patient was exposed to the test treatment. After that, then what should occur is a question about what the risk benefit is to continuing that patient in the study, even if they are pregnant, and to reconsent them in that process. In other words, once you know if the patient's been treated, then they might be able to be continued in the study, at least for safety, even if we have to break the blind. But that requires the consent of the patient. So one of our best practices that we describe in the clinical ethics guidance that we have on pregnancy is to reconsent that patient and to continue to follow them on treatment if that's appropriate. And in addition, one thing that I think is very important is to collect PK data on that patient, because the PK data can help us understand better whether or not there needs to be dosing adjustments during pregnancy. Now, the second part of that question is what about the fetus and what about the infant after it's born? So again, depending on the situation, for example, with biological agents that are immune modulators, particularly if they're given in the third trimester, can pass and cross into the fetus through the placenta. And that can potentially have effects on the immune system of the newborn and infant. So depending on the drug that is under study, there might be a need to follow that infant, not just after birth, but for some period of time after birth, even and including up to a year or more. So we do have information about that in our pregnancy clinical ethics guidance related to pregnancy that I can direct you to. But it's a very good question. And there are a lot of things that can be done besides just saying that the pregnant individual has to be removed from the trial and not followed up. Thank you, Leonard.
Thanks very much, Lynn. I think an excellent explanation, emphasizing really the importance of getting the maximum amount of information about these situations that happen during the course of a clinical trial, both in pregnancy and obviously in exposed neonates. Shabnam, a question for you. Here, since an AE can include a worsening of an existing condition or may be new but appear gradually, when should the beginning of the AE be considered insofar as trial records are concerned? So I guess the question here is related to when do we regard the adverse event as beginning in relation to when the trial, the patient was put on the drug?
Okay, thanks, Leonard, for the question. So the adverse event definition, just recap that it's any untoward medical occurrence associated with the use of a drug in humans, whether or not considered drug-related. And adverse events that occur during the study drug treatment should be coded as treatment emergent to differentiate from events that may have occurred prior to study drug treatment. I think that's important concept. Treatment emergent adverse events, they're coded in MEDRA with a flag, usually called a treatment emergent flag, and that would distinguish them from other AEs. For example, perhaps one that may have occurred right prior to study treatment, perhaps a worsening of a condition even prior to receiving study treatment. If we're trying to distinguish AEs which may have been related to worsening of an existing condition, that did occur on drug, you may consider coding it in such a way that it reflects actual worsening of that event. So I hope that's clear. Over, Leonard.
Thanks very much. Okay, I have another one for you, Lynn. Would it be appropriate to recruit participants for studies to develop drugs for diseases that impact people from minority populations or children differently than drugs being developed for the general population? And the example that's given is this might be at hospitals, clinic, or disease support groups. So I guess a question targeted at drugs for diseases that impact people from minority populations. Any thoughts about that?
Yes, thanks, Leonard. An excellent question. One of the things that we have learned, particularly through that ADEPT 9 workshop that I mentioned in my talk, Enhancing Diversity in Pediatric Clinical Trials, I think it's right to state that, and as I mentioned in my talk, that, we need to study drugs in the populations that we believe are taking them. And so what we don't want to see necessarily is a drug development program for a disease that includes a substantial number of minority populations, that population is only studied in clinical trials that, for example, enroll a very non-diverse population. And I'll tell you why that's important. That's important because what we heard at this conference, this workshop that we convened, was that not only is it important to understand whether the drug behaves differently in a minority population compared to a non-minority population, but that when patients are recruited from distinct areas that have generally been underrepresented, that enrollment of those patients gives trust in those communities that this trial is worth something, and this drug, if it's approved, is actually acceptable to take. We heard from many stakeholders, including patients, minority and underrepresented populations, who said that, why would we take a drug that was approved by FDA? Because we don't trust the government, and nobody in our neighborhood even entered the clinical trial. But if there's enrollment of patients from those underserved areas, and those patients go back and talk about their experience, not only does it help with recruitment in those areas, but it helps after, as I said, the drug is potentially approved and available, that it gives confidence and trust that those drugs are actually safe and effective, as FDA has determined that. So yes, I agree that it's important to enroll a diverse population, including those that might be historically underserved. And yes, those sites can include regional clinics. They can include engagement with disease support groups. And again, that idea of the decentralized trial is also one that can help with enrolling populations that have been historically underrepresented. Thank you.
Thanks, Lynn. I think you make an excellent point that inclusion improves trust in communities that are not generally participating, a really important consideration. Shabnam, I have a question for you. The question's simple. I think the answer has nuances. What kind of study will have death as an endpoint? Do you want to respond to that one?
Yeah, sure, Leonard. Thanks for the question. Yeah, what kind of study would have death as an endpoint? Death is an endpoint for several types of clinical trials, and especially for diseases that are considered serious and life-threatening, it can be very useful. An example of that would be assessment of an all-cause or a 14-day mortality, which can often be an efficacy endpoint. Again, in trials that are looking at conditions that are serious or life-threatening. And in the anti-infective world, one example could be a phase 3 trial for example, for a healthcare-acquired or healthcare-associated or ventilator-acquired bacterial pneumonia, in which we often have efficacy endpoints looking at mortality at a certain time point to assess the non-inferiority of one antibacterial drug versus another standard of care antibacterial drug. And then in the talk, as you recall, the example was for an antihypertensive drug that was looking at endpoints related to mortality and acute myocardial infarction and stroke and that sort of thing.
Yeah, thanks very much. Sorry, Shabnam.
Yeah, that's it. Thank you.
Yeah, I just had some thoughts as well. Outside of the realm of infectious disease, obviously cancer trials are very often have mortality as an endpoint or time to death, because many of those patients do experience death. The other thing that probably is just that came to my mind was sometimes it's difficult to tell whether death is the endpoint as a result of a failure of the drug or whether it may be some other cause. And, I think sometimes evaluating death may be tricky, particularly if patients have serious underlying comorbidities. So I think it's a very insightful question. Let's see. I'd like to move on to another one for Lynn. This one's about dosage extrapolation, Lynn. The question says, we can't extrapolate dosage in neonatal populations because of the very different physiology related to gestational age, right? Any answers to that?
Yes, I do, Leonard. Thanks for that question. Again, another very insightful question. So it is true that, and again, in my discussion of extrapolation, it relies on how similar the population is with the disease, a particular pediatric one, compared to, for example, an older pediatric population or an adult population. And we certainly understand that neonates have a very rapid change in their physiology, their metabolism, their physical growth over a very short period of time. So it is hard to imagine that neonates could be similar to an adult population, for example. So we'd have to absolutely do separate neonatal trials. And the answer to that is that, it may be true that trying to compare a neonate to an adult would be difficult, but comparing a neonate to, for example, a 6-month-old or a 3-month-old, and if we have dosing information and efficacy established in those groups, it may be very reasonable not to have to conduct, a fully powered, randomized, placebo-controlled trial in the neonatal population, but leveraging what we know about that 2-month-old or 6-month-old population, that we would still need to collect probably safety information, and yes, to your point, dosing information, because we recognize that those metabolic enzymes develop rapidly during the neonatal period. So yes, we would likely need to have dosing information, and we would likely need to have safety information, but probably not necessarily in all cases separate neonatal randomized controlled trials. And the only other point I want to make too is that if, for example, this drug had been tested in neonates for a different condition, and now the same drug is being looked at for yet another indication in neonates, then having that information from another previous development program in neonates can be very helpful, and that information could potentially be leveraged. Thanks.
Thank you very much, Lynn. As a non-pediatrician, I can only imagine how difficult it is to study neonates for the activity of new drugs. Shabnam, I have another question for you. How is the term severe used in the classification of AEs?
Yeah, thank you for the question. I'm glad the question was asked because this does come up a lot. And so, I had mentioned in the talk that severe does not equate with serious, as that is a regulatory definition. But severity can be an indication of intensity of a specific event. So that's the key word, is the intensity when we're talking about how severe something is. Severity can be assessed by those toxicity grading scales that I had mentioned. And you can, reference some of those examples on the slide just to take a look at what may constitute mild, moderate, and severe for different patient populations. So in general, severe AEs would, as you would think, would be those that would cause significant pain, discomfort, for example, or may prevent daily activity, as an example, depending again on the study population. What may be severe for one patient population, like a healthy volunteer, may not be the same for oncology patients, for example. And then in terms of laboratory abnormalities, a severe AE would probably be one, obviously that's worse than mild and moderate in terms of the actual normal reference ranges, and it might be one that might require medical intervention. So I think hopefully that is clear. Thanks.
Thanks very much. Again, let's just alternate. Lynn, a question for you about pediatric extrapolation. Thoughtful question here. For pediatric extrapolation, can you use modeling and simulation before starting your trial for a better informed program?
Yeah, thanks, Leonard. That is a really good question and a very important one. So as I mentioned, extrapolation, this idea of extrapolation is to leverage what we know about a reference population, an adult population or older pediatric population, and apply it to that population that you're interested in studying, which is usually a younger target pediatric population. And the whole notion of modeling and simulation, that is to take data that we have and to understand those relationships within the data using computational methods. That idea of modeling and simulation is a critically important component of the analysis of data. Not just, people say, well, of course we do modeling and simulation to derive a dose. If we want to model a dose in children that could match an adult dose that was effective. Yeah, that's clearly something that we have and that we do in even overall drug development. But one of the things where I want to point out another important place where modeling and simulation can be useful is in evaluating the data that we have that even supports similarity between an adult and pediatric population. So you may have data from different published studies. You may have data from case studies. You may have data from registry studies, and you may have animal data, for example, that can help be used to support a similarity of the disease between a pediatric and adult population. Well, these types of data and using modeling are tailor-made to understand the relationships. So I think that this is a great way to use modeling and simulation. In addition to the uses, once you have established an extrapolation concept and want to figure out dosing, etc., as part of a plan. I'm sorry, I'm losing my voice, but thank you.
Thanks very much, Lynn. Are you up to another question? We'll see how your voice holds up. But this one,
I just took a drink, so I think I'm okay.
Okay, I'm glad you recovered. So here we go. I think this one it's a great question because it opens up a couple of important points for discussion. So what are some ways to address challenges in conducting clinical trials in pediatric patients? For example, how can you obtain sufficient information when the pediatric population available for the study is very small? Any thoughts about that?
I do. And in fact, we convened a workshop 2 years ago on innovative trial designs that specifically could be considered as part of a pediatric drug development program. And by the way, the reason I think that this topic is so relevant, even if you're not interested in the pediatric drug development per se, is that the same principles that apply to developing drugs for small populations also applies to rare diseases. So many pediatric diseases are considered relatively rare compared to the adult population, just like rare diseases overall. So the same principles and the same strategies and the same innovative trial designs that we use in pediatric drug development can be applied in many cases to rare disease drug development. So the idea of Bayesian approaches, the idea of, externally controlled studies, the idea of N=1 trials, all of these potential strategies, and of course the use of modeling and simulation to help decrease the number of patients that you might need. All of these approaches are discussed in the ICH E11A guideline that I described to you and is available now on the website for ICH. So it's really important to consider, and we have many different approaches, and that guideline, I think, really provides excellent examples and references for you. And again, I would encourage you to read that, because if you're interested in rare disease development, some of these same study methodologies and innovations also apply there. Thank you.
Thanks for a great answer. I guess emphasizing that one really needs to be as creative as one can at getting the information that's needed. We're running close to the end of the session. Shabnam, I have one other for you. I think this is regard to safety information. What type of information would go into the investigator's brochure?
Okay, thank you, Leonard, for that question. So the investigator brochure or the IB should have in it all the information that's important for the investigator who's administering the drug to human subjects to really know and understand. And it's required that the information include information on drug substance and formulation and the pharmacological and toxicological effects of the drugs and then again in humans if known. And then obviously information relating to safety and effectiveness in humans obtained from prior clinical studies, and that's going to be key. Also, possible risks and side effects that are anticipated on the basis of prior experience with the drug under investigation, and then also experience that are you can have from related drugs in the same class, for example. And then lastly, it should include precautions or special monitoring to be done as part of the investigational use of the drug. I think there's another talk on investigational brochures tomorrow as well. Thank you.
Great. Yes, I think some of that will be covered. Just one more for you, Shabnam. Interesting question. In the hypothetical case, does the investigator report death or anaphylaxis or both? I presume if the patient has an anaphylaxis and dies, how does that get reported? Any thoughts about that, Shabnam?
Yeah, thank you for the question. So, the investigator would report to the sponsor this AE as a death, and the cause of death, should be clearly communicated as due to anaphylaxis. In this case, death by itself would not be very informative without letting the sponsor know what they think is the cause of death. And in addition, a causality assessment by the investigator should be provided to the sponsor. As much information about the event as you could note would be helpful to the sponsor, who would then ultimately determine when and whether to report to FDA in this hypothetical example.
Great. Thanks for the answer there. So I think that pretty much brings us to the end of this session. Again, a vote of thanks to both of you for excellent talks and Q&A session addressing safety and addressing specific populations. I think it brings to the close our content for today, which is really focused on clinical trials and what happens in clinical trials. Tomorrow we'll reconvene, and I think the topics that we'll begin with are non-clinical and early clinical drug development, which we haven't discussed yet. They include chemistry and manufacturing controls, pharmacology and toxicology, and clinical pharmacology, all very important topics that obviously we base our design of clinical trials on. Following that, we'll have a couple of lectures, one on ethical considerations, which we haven't touched on yet. I'll be giving a short lecture on innovative trial designs and approaches, and then we'll finish off tomorrow's sessions with two talks on very innovative treatment modalities that are just coming to the fore and offer a lot of promise for diseases that we really couldn't deal with before. So an exciting lineup for tomorrow, and I'm very much hoping that everyone will join us again.